Hello, everyone. I'm here to talk to you today about a line of research which Dr. Azad and I have been pursuing uh, that has allowed us to create a predictor of ACE2 binding ability in Betacoronaviruses. If after today's talk you'd like to learn more about our method or our findings, you can access this citation here at the bottom, which is the paper that we've published on this method. So, within the last 20 years, three deadly human coronaviruses have emerged. And of those, two-thirds of those have been what we call SARS-like coronaviruses. So they're similar to SARS, which is SARS-CoV-1, essentially, which was the original SARS virus. And uh, those same two-thirds use the ACE2 receptor. So not only is the temporal scale here very important, because within the last 20 years we've had three deadly coronaviruses emerge, when before that we have no record of a deadly human coronavirus. So we're at kind of a veritable explosion of emergence of deadly coronaviruses in the human population. Uh, So SARS-CoV and SARS-CoV-2 are very closely related. In fact, so closely so that they're considered the same species. But when you look just at their receptor binding domain, the part that binds to the ACE2 receptor, using multiple sequence alignment techniques, you find out that the receptor binding domain is not nearly as closely related as you would assume based on these two viruses being the same species. Um, so, for that reason, it's been proposed that recombination could account for the emergence of both SARS and SARS-CoV-2, as a matter of fact. Um, so, this could account for this explosion in the number of deadly human coronaviruses. However, uh, recombination regarding the receptor binding domain and the ability to bind ACE2 does not necessarily have any effect or any reflection upon the emergence of MERS, because it uses a totally different receptor. So something's going on here that is triggering coronaviruses to emerge in the human population. Um, us, along with basically, half of the scientific community out there that's studying the evolution of uh, Betacoronaviruses, are looking for what triggered this. So, and trying to contribute to what triggered this explosion in the emergence of deadly human coronaviruses. Mainly because we need to predict this. We need to stop it. We need this not to happen again. So, our technique that we're presenting today, or that I'm presenting today, is one way of predicting and monitoring the emergence of receptor binding domains, or coronaviruses which contain receptor binding domains that could bind to the human ACE2 receptor, and therefore could become deadly human coronaviruses. In addition to recombination, it's also possible that just as the result of opportunity, having the opportunity to develop the ability to bind to the ACE2 receptor, that coronaviruses have evolved uh, separately to bind the ACE2 receptor. So this would be convergence. So convergent evolution could also account for the surge in deadly human coronaviruses. So for that to be the case, there would have to be conditions that exist that were not existent before 20 years ago, that promote the convergence process or convergent process. So one hypothesis for this is that it's just greater opportunity as a result of these wet markets in China. You know, humans are regularly exposed to coronaviruses there as a result of the animals that are brought into the market that likely carry different coronaviruses at different times. The third option, for the reason for having a couple of different lines of uh, receptor binding domains in deadly human coronaviruses, specifically SARS-CoV and SARS-CoV-2, is that there was a ancestral coronavirus that had the ability to bind ACE2 that resulted in both of the lineages that SARS-CoV and SARS-CoV-2 respectively belong to. So that's kind of the problem that we're trying to get at is more can we predict the ability to bind ACE2, but our dendrograms also provide insight into whether it's common descent, uh, convergence, and or recombination. And it should be noted that those aren't mutually exclusive. You could have common descent with periodic recombination, which actually, if it's fast enough, becomes gene conversion. Uh, so there's no reason why all of these should be considered mutually exclusive, or these three should be considered mutually exclusive. So standard phylogenetics, as I said, results in two different clades of ACE2 binding receptor binding domains. And one of these clades contains the original SARS virus here, and one of them contains SARS-CoV-2. But they're separated by 49 different taxa, 49 different coronaviruses that are not ACE2 binding fall between these two clades. And in addition to that, we don't have a great level of support for these clades altogether. So it's entirely possible that what we're looking at here is the incorrect or wrong topology with regards to receptor binding domains and how they evolved in the beta coronaviruses. So, uh, so with regards to evolution, selective pressure applies to traits. So we're going to want to look at how these sequences evolved in order to kind of try to unravel the mystery of how ACE2 binding evolved. So we got to take a little bit of a step back and really think about how evolution occurs. Evolution occurs by selection for certain advantageous traits. And sometimes we lose sight of the fact that these are traits. These are not uh, different residues at different positions in the amino acid sequence. These are not necessarily the different nucleotides at different positions in the nucleotide sequence. These are what those result in, the traits that they result in. And uh, those traits are functionally specific. So the trait here would be the ability to bind ACE2. And the reason it might be selected for would be because it would be more advantageous. The virus would be better able to replicate and persist if it could enter a human host. So, multiple sequence alignment algorithms, they really kind of come at that, the fact that traits are functionally specific from a different point of view. They come at it from a probabilistic point of view. So they say, okay, we got this group of proteins here, and we know that they're related. Okay, so let's see what the probability of getting a change in a cysteine to a tryptophan or an alanine to a lysine is. And they come up with scoring matrices. And those scoring matrices are then used to, uh, align sequences and look at how homologous sequences compare to each other uh, using an alignment created from those probabilistic scoring matrices. Um, most commonly observed substitutions are based both on preservation of function as well as ease of transition. Now then, when you look at the probabilistic scoring matrices, you're looking uh, really more at the ease of transition, because if you have a amino acid that is very functionally similar to another amino acid, but yet it requires the substitution of all three nucleotides within the codon that encodes it, then that's not going to be a very probable substitution because it requires a whole lot of change to get there. But when we start looking at the functional uh, conservation of or conservation of functional groups, uh, then we start looking at greater lengths of evolution. We look at evolution where we're not necessarily as concerned with the ease of transition because over greater lengths of time, uh, we can change all three nucleotide positions and still maintain the physical chemical properties of the amino acid or close to it. Uh, so there's certain instances where preservation of function uh, does not necessarily go along with ease of transition. Um, so as I said, this ease of transition becomes less of a factor as the amount of evolution increases. So this is not necessarily the amount of time, because if you got a section of DNA or within a virus or whether you have a another section of DNA within another population, let's say mammals, and you look at from mammals to archaea, that's a great amount of evolution. Well, if you look at that little section of DNA in a virus, you may see that same amount of evolution over a year or 2 years, a very short period of time. So it's really more related to the rate of evolution than it is to temporal time span. So therefore, functional preservation, not probabilistic observation, may better resolve homologous relationships over great amounts of evolutionary time. So, in other words, we need to look at the preservation of the selected for physical chemical profiles of a protein rather than looking at the probabilistic alignment between two homologous proteins. Now, the probabilistic alignment is very good as long as uh, the amount of evolution is not great. So what we're getting into here is what is called the twilight zone of homology, and that's less than 30% uh, identity between two amino acid sequences, uh, but yet we have homology. So these are homologous sequences that show less than 30% amino acid identity. So that's the twilight zone. And the methods which we're developing really attempt to reach into the twilight zone and resolve those relationships. So I've mentioned a little bit about physical chemical properties. So amino acids, we talk about them with regards to their single letter or 3-letter code. Uh, and with regard to alignment, we look at substitutions. But the real world is that there's redundancy with those amino acids, because if you look at just their physical chemical properties, there's overlap in those physical chemical properties, and that's what we generally refer to as conservative amino acid substitutions. You replace one amino acid with another that's very similar to it physical chemically. Um, there's several different physical chemical properties. So, one amino acid may be similar to another amino acid for one of those properties, but very different from it with regard to another property. So, we'll talk about it just a little bit more here in a little bit. But when you start looking at the conservation of physical chemical properties and applying it to homology determination in proteins, you really got to know a little bit about what the function of the protein you're looking at is, or even the domain of the protein that you're looking at. So in our case, we were looking or are looking at the receptor binding domain of Betacoronaviruses. So we know that this receptor binding domain main function is to lock together with the ACE2 receptor and allow entry of the virus into human cells. So we know that it's a protein-protein interaction. So we can kind of choose our physicochemical properties based on that. And in fact, the two that we chose, which we'll talk about shortly, but molecular weight and hydrophobicity are very key in protein-protein interactions. So along this same line of thought, this was explored back in 1998 by Lolkema and Slotboom, and they made the statement that three-dimensional structure of amino acids, presumably, is quite tolerant of changes at the amino acid level. So basically, in that one little statement right there, they have included all of the redundancy that we just talked about with regards to physicochemical properties. Three-dimensional structure is quite tolerant of changes at the amino acid level. Okay, that redundancy does not necessarily mirror the changes that we would expect based on ease of transition. And we'll talk about this a little bit more in a second, but you can have changes that are very conservative with regards to physicochemical properties, but that result in lots of change or are the result of lots of change at the nucleotide level. So really lots of evolution. Localized functional specificity. So I touched on this a little bit just a second ago, but every part of a protein does not have the same function. So while you can use probabilistic models to align amino acid sequences without really having to know what function a particular part of the amino acid sequence has, you can't do that when you're looking at physical chemical properties. You got to know a little bit about what's going on there and what it's doing before you can really focus in on that part of the protein. So physical chemical property based analyses are necessarily constrained by localized functional specificity. So that's basically just saying you got to know a little something about what you're looking at. So there's two physical chemical properties that contribute the most to the three-dimensional protein shape and have been considered fundamental um, with regards to protein-protein interactions. So since what we have here at the binding of the ACE2 receptor to the receptor binding domain of the Betacoronavirus, since what we have there is a protein-protein interaction, uh, we chose molecular weight and hydrophobicity as the two physical chemical properties that we would analyze with regards to that domain. So using those two physical chemical properties in conjunction with each other, we were able to come up with a metric that allowed us to look at the distance between two receptor binding domains uh, functionally based on physical chemical properties. So this is all built upon, or it all builds upon the concept that the complementary shapes of two proteins, that they must have complementarity in order to interact with each other. All the other physical chemical properties are more for specificity. Once we have the complementary shapes, we have to have the complementary shapes, and then the other physical chemical properties, other than molecular weight and hydrophobicity, can come into play. But if we don't have these two fundamental physical chemical properties and conservation of them, then we're probably not going to have conservation of function. So that's why we chose molecular weight and hydrophobicity. So this is just a little chart I put together here that shows the overlap uh, of the different physical chemical properties that we looked at. Now we had molecular weight and hydrophobicity. We came up with a metric which includes both of them in the same metric. Uh, But you can also look at them separately, molecular weight and hydrophobicity, as is done on the left-hand chart here and in the right-hand chart here. So whenever you look at it that way, we see a lot of mix matching with molecular weight versus our metric. But hydrophobicity pretty much corresponds directly across uh, from our metric to hydrophobicity, with the exception being the highly hydrophobic and very large, or very high molecular weight amino acids. There's a little bit of mixing up there. So there is additional information that can be gained by combining molecular weight and hydrophobicity that is not necessarily contained in just hydrophobicity. And in fact, we've ran some analyses that have confirmed this, that hydrophobicity alone does not reveal as much information as our metric does. So the assumption that we made was that given the overlap and thus redundancy of the physical chemical properties, the maintenance of a function or the conservation of a function will favor the selection of amino acids that are closer on the continuum that we just looked at rather than farther away. So that continuum is based on just the two physical chemical properties of molecular weight and hydrophobicity, but it combines the two. So, for example, if we look back at the chart, which I'm not going to scroll back to it, but we would find that a methionine to tyrosine or tyrosine to methionine substitution is actually a conservative substitution, despite the fact that it requires the change of all three nucleotides within the codon. As far as the way that this was applied back in 1998, Lolkema and Slotboom, they looked at primarily just the content of the amino acid sequence. So they would take a particular amino acid and they would assign it a hydropathy category as internal, external, or ambivalent, represented by I, A, or E here. So they would convert it into these strings that it was were just a combination of I, E, or A, I, A, or E, either one. And so that accounts only for content. This particular amino acid, let's say that it's a leucine. Well, if that's a leucine and that's a leucine, it's still an A, regardless of where it is in the sequence. So they have counted only for content. The solution is to encode waveforms in a way that accounts for both content and context. So this provides a whole lot more information because, you know, at any given point in a amino acid sequence, whenever it folds into its final structure, in some cases, maybe you just need a linker, maybe you need a loop there or whatever you need there, but that might be able to be achieved by one or a couple of different amino acids put together. So we need a little bit of context. We need to know a little bit about the stuff that's surrounding the amino acids that are surrounding a particular amino acid. So our method in encoding waveforms, which this is just one waveform here. So this is an amino acid sequence, and all I'm showing here is this arginine. It has a positive value here, but yet because of the context, it flips all the way over here to negative over here. So just based on what's around it, it gets a totally different value. And you can see this too here with the phenylalanines. You can see that each one of these phenylalanines, which are several here, has a slightly different amplitude. So our method of encoding these waveforms accounts for both content and context. So that's very important. Now then, we need a way at this point, or needed. Now we've got this all worked out, but we needed a way at this point to compare the physical chemical profiles as they were encoded in waveforms in order to develop a distance between two proteins, between two waveforms. So what we came up with and what we use is dynamic time warping. Is what dynamic time warping does is it stretches or compresses the x-axis in order to align two waveforms. And the way it does this is it minimizes a distance metric or distance measure. And in this case, we're using Euclidean distance. And it minimizes that Euclidean distance And once it finds the minimum of the Euclidean distance, then that's the best alignment. So it stretches and compresses the x-axis basically iteratively until it finds the best alignment based on the least Euclidean distance. So just to point out one other little thing here while I have you on this slide, these are two amino acid sequences that are actually only 21% identical, but yet they have an almost identical waveform. So what this would represent is a potentially a protein that is way into the twilight zone, has very little uh, identity between the two homologs, but yet functionally they could perform the same function. In fact, these here are the bottom one is a synthetic construct that I made, and I think we talked about in the spring, in my update this spring. But it's a synthetic construct that is only 21% identical to the top one, but yet both of these sequences, if you predict the structure, are almost identical. So not only are their waveforms identical, but the waveform uh, correlates with actual modeling of the structure of this sequence, and both show that these functionally or structurally these two amino acid sequences are very similar. So the distance between the two proteins is the lowest Euclidean distance, as I just said, after DTUW. So this is after DTUW. It's actually a part of DTUW is minimizing that Euclidean distance. Once we have the distances among all pairwise combinations of proteins that we look at, so we have 10 of them, we can look at all the pairwise comparisons there. And once we have that distance matrix, then we can use hierarchical clustering to construct dendrograms, basically, to look at how these sequences are related to each other topologically. So in our study, we use UPGMA and neighbor joining to look at the sequences, or to look at the dendrograms for the sequences. So we got a surprising result, and this is really what led to our publication and the focus of our publication. So as we talked about earlier this morning, there's two different groups of ACE2-binding Betacoronaviruses. Whenever you look at them using the widely accepted maximum likelihood analysis using multiple sequence alignment. So, and if you remember right, I said there was 49 taxa, or a number of taxa, 49 in this case, between the two clades of ACE2-binding Betacoronaviruses. Well, when we look at this from a physicochemical profile and we use hierarchical clustering with our waveforms, we find that there's actually only one clade of ACE2-binding Betacoronaviruses, and the 49 taxa that were in effect between the two clades of ACE2-binding Betacoronaviruses are no longer between them. And not only that, but all of the ACE2-binding Betacoronaviruses in here have been confirmed as ACE2-binding Betacoronaviruses, with the exception of 4 pangolin sequences that have been highly suspected of being ACE2-binding, but have not yet been confirmed in a lab to bind ACE2. Essentially, they just haven't been looked at yet. So this is very important because let's just go back to this maximum likelihood tree here. Okay, let's roll back to before COVID-19, before we even knew that there was a virus coming. If someone had been out there monitoring for the ability to bind ACE2, and a sequence had come up They were using amino acid sequences to monitor for ACE2-binding Betacoronaviruses that belong basically to this clade right here. Okay, uh, they would have assumed that a sequence that ended up in this clade here, that now we know contains SARS-CoV-2, uh, was really not a threat, because they would have said, okay, it's not really all that closely related with the receptor binding domain to SARS-CoV, and SARS-CoV is the one that we know that has a pretty high death rate, and it can affect humans. So if it was in this clade, they would assume that it really wasn't that big of a threat. Okay, now we know that it is a big threat. So yeah, we could monitor for sequences that uh, cluster with either of these clades, or found in either of these clades, using maximum likelihood, and we'd probably have a pretty good chance of predicting the uh, emergence of another uh, beta coronavirus that could bind ACE2. But before we knew that SARS-CoV-2 was in that clade, we wouldn't have been able to do that. Now, fast forward to the implementation of our technique. Anything that falls in this clade here would have been suspect with regards to ACE2 binding ability, and perhaps the ability to emerge in the human population. So it's much more specific, and in being more specific, is perhaps more reliable, and it would be better used as a predictor, a monitoring tool to monitor the emergence of ACE2 binding beta coronaviruses in the human population. Another thing I might point out here is, so there was 119 sequences in our analysis, and I used the IML web server to calculate the maximum likelihood tree. That took about 42 hours for these particular sequences. Um, Using our technique, we did it with R in about 15 minutes. And we'll talk a little bit about the overall topology and how they compare coming up. So these are the whole. This is the whole data set of the 119 beta coronaviruses, and in the center here you can see the two different clades of ACE2 binding beta coronaviruses. And then you can also see the 49 taxa over here. Uh, so this is the maximum likelihood tree, and you can see that they're separate over here. If you follow these, you can see the 49 taxa are actually between the two groups of ACE2 binding beta coronaviruses. And then we can see over here that we actually only have a single well-formed clade. So basically, this is the same tree that we've already looked at. It's just not it hadn't been condensed. So, and this is a tangogram showing the two trees. It shows our technique over here and the maximum likelihood technique over here. And the reason why I included this is because it was the analysis of these tangograms that revealed that we were actually getting more information from our technique, potentially than the maximum likelihood analysis provided. So we were seeing that, hey, this functionally confirms that our technique is able to uh, cluster these beta coronaviruses, or RBDs from them anyway, into a functional cluster, and it's a single functional cluster. So you can infer from that that this cluster may have only evolved once. It may not be that it evolved twice. The other thing I wanted to point out here, which is something that we haven't really explored a whole lot, but when you look at these tangograms and you see uh, clades that are somewhat distantly related via the tangogram, they're not as close on the maximum likelihood side, and then functionally they come together on the molecular weight hydrophobicity side with the dynamic time warping. Uh, this could also be an indication of convergence, because you got two different groups of sequences here that, when you look at them based on the actual amino acid sequence, they're more distantly related than if you look at them based on the physicochemical profiles. They may have actually just converged upon that. So our technique doesn't necessarily rule out convergence. So the other way that we looked at congruency between the different trees was we looked at the common nodes. So this is basically a comparison of the nodes that have the exact same terminal taxa. So essentially what you would say here, if we were looking at a comparison of maximum likelihood to neighbor joining, so this is multiple sequence alignment based maximum likelihood, multiple sequence based neighbor joining, 77% of their nodes contain the same list of terminal taxa. This is important because our DTW based analyses were only slightly less than that. So you got 77% that contain the exact same list of terminal nodes or terminal taxa with regards to neighbor joining versus maximum likelihood. You got 76% with regards to our DTW based UPGMA or neighbor joining analyses. The third way that we looked at congruency between the trees was we used the Robinson-Folds metric. So the Robinson-Folds metric, whereas the common nodes looked at basically clade composition, the Robinson-Folds looks more at the overall topology of the tree. And when we look at the overall topology of the tree, we find that the multiple sequence-based maximum likelihood and neighbor joining we're only about 53% similar. We actually get a greater amount of similarity whenever we use our techniques, so 54 and 58% respectively. So some conclusions. Parsimony and our analysis supports one clade of ACE2-binding coronavirus. It's just a violation of parsimony to say that the ability to bind ACE2 evolved, at least through descent, more than once. So while we can't rule out convergence, evolutionary descent seems more likely at this point. Clade composition is largely conserved using our method, as conserved using our method and comparing it to maximum likelihood as it is between the multiple sequence alignment based techniques. So essentially what we're saying there, what I'm saying there is that there's no reason to suspect that ours is less accurate than, say, neighbor joining, which is another very accepted phylogenetic reconstruction technique. So overall, the topology is more conserved, actually, between the maximum likelihood and multiple sequence-based maximum likelihood in our technique than it is between the alignment-based techniques. So we're actually recovering more of the maximum likelihood topology with our method than you do using multiple sequence alignment-based neighbor joining. So some future directions where we're going to go. We need to carry this to a point where it's a little bit more intuitive for the average biologist out there. And one of the ways that we can do that is to use a waveform guided standard alignment. So instead of looking at waveforms, which represent physicochemical profiles, we can align amino acid sequences and show their multiple sequence alignment, or at least a pairwise sequence alignment, using the waveforms as a guide. We also, actually, this is currently going on with an undergraduate in the lab, investigate other groups which could have seeded the ACE2 binding ability into the beta coronaviruses. So at about 20 years ago or 20 plus years ago, something happened. Something happened which has kind of triggered the emergence of multiple deadly human coronaviruses. We need to know what that is. And so one of the potential reasons for that could be that another group of viruses recombined with coronaviruses, resulting in this ACE2 binding receptor binding domain. And we're actually seeing some pretty interesting stuff with that, looking at different families of viruses. And other than that, at some point, it's going to be time for me to get back to applying this technique to prions. So that's where we're at right now. That's my lab update. And if you have any questions, I'd be glad to take them outside of this talk. I had to record this talk because of my current situation as far as internet. But I can take questions, and I will be in the meeting live. So if you have any more questions or any questions, just let me know.